So for the whole chapter, we're gonna, have an overview, go through the general structure, how they classify them, just like what we've done with other groups of organisms. We look at the viral multiplication, and it's pretty interesting. Once we look a little bit more into how how the hereditary material is read, how the chromosomes and how the DNA are translated into proteins, and how the viruses. We're gonna get an appreciation for how viruses actually take advantage of that system. It's almost like, because they're so small, I always imagine them as the smallest possible, which it might not be the smallest, but it's the most sufficient or intelligent way to do things, because they really have almost nothing. They just let host cells take over the production of whatever they need, the few little things they're built out of. So the host cell reproduces their genetic material and maybe some of the proteins that they need to cover that, but that's really it. This already is this is called a bacteriophage, which is a virus that actually invades bacteria, and so this is already a really complicated structure in comparison to some of the other viruses. So this would be falling on the multiplication of bacteriophages. And then how to culture and identify viruses, and then at the end there's going to be some talk about infections detection and treatment, but we're not going to get to that today. So what do you think was, if you think of the few microbiologists we talk about, around what time was the first microscope? 1774, is that somewhere like that? Earlier? Anyway, so what do you think, say it was 18th century sometime, was the problem, how were they able to detect, the first shapes and arrangements of bacteria but not know anything about viruses?
Viruses are small.
So they couldn't, even with today's light or microscopes that use visible light as a source, you wouldn't be able to really see them, and so it took a while. But what they did find is that when they used certain methods to get rid of all the bacteria, they were still able sometimes, or in certain cases, to cause infections. So they knew there may be something else, and they knew that there must be a smaller organism that causes certain disease. And Pasteur was one of the first ones to postulate that rabies may be caused by what is now called or known as a virus, but then just a particle that is smaller than bacteria. And these two showed that the disease in tobacco was caused by a virus in the late 19th century, and by 1950 it really was more established, and a lot of research was done to try to figure out what the structure of those are. Yes?
When did they start being able to see them?
It's a good question. It probably depends on when they first saw. Either the size of the size varies, but I think the main support for the theory of viruses came really from experiments, not from visualizing them. So yeah, I don't know when they first started seeing them.
Got medicine too.
And so now they're defined as non-cellular particles with a definite size, shape, and chemical composition. So we know a lot more about them now through extensive research, starting in that area. There's no real agreement on how in evolution, where they originated, where from, and when. So there's no real idea of how that happened. They're considered the most abundant microbes on Earth, and I think I read about, it might be in the book, in a milliliter of water you could expect millions of them. So it's amazing what's in there, and how ubiquitous, but also how dominant they are. And you can also then imagine that in terms of, when we hear virus, it's usually considered a negative organism or a negative thing, and there must be a lot of them out there that have no real effect. And there's also the option, the possibility that they do a lot of beneficial work for any kind of other, protozoan or animal. I mean, eukaryotic or eukaryote, prokaryote, for any kind of higher organized organism we might not know about. And mainly, what I want to point out is they're obligate intracellular parasites, so they're not living out of a host. That's one of the main important things also, that they do not have a cellular structure. We talked about this enough. You can look through this table, but you'll probably see things like the basic structure, the capsid, etc., and the nucleic acid core, DNA or RNA later on. So that's just a summary of all the characteristics of viruses. So we talked about the size, yes?
You said they're not living out of a host. That means they cannot survive outside the host?
Well, that's you can have some survival, but they're not reproducing. There's no reproduction possible outside the host. There's no, they can't reproduce and live outside the host cell, but they do survive certain amount of time, depending on what kind of virus it is outside of a host cell. So if you get infected through with a virus, a lot of times there are phases between where they're outside of the host cell, but they won't be able to just live outside the host cell and reproduce and then go back, for example. So the size in comparison to what we've looked at, for example, Streptococcus is one of them. Looked at E. coli a lot too. That's the size of them, and you remember how hard it is to just see those. So if you look at some of the viruses or some of the shapes that you find, and you can see what the difference is again between these and these. So if back to the question when they were first discovered, this one is not that far off. It might have, depending on what they were looking at, probably determined how quickly they were actually seeing those. The other question is, when did they start identifying those as not being bacterial cells but viruses? And some of the famous ones, HIV's in there, number 4, And we'll talk about like what their structures look like, or what the differences in structures are. If you think back to the first picture, the bacteriophage looks completely different, like a lot of those. So you have one over here, for example, but that looks different than that first picture too. You've got a lot of them that are, they seem to be having this outer layer around like a certain structure in here, a capsid structure, same thing here, so there's kind of like an envelope, and some don't have that. Like here, this one doesn't have that envelope, but has these spikes. So we'll talk about the difference between those. But they're pretty fascinating overall because, like I said, they're very efficient. There's no need for, not even for a cellular structure to regulate what goes in and what goes out, and to at least have, for example, ribosomes and produce your own enzymes, and then with those again control in and out and nutrient uptake, etc. That's all not existing in bacteria and viruses. So just remember that. So they lack, which I just mentioned, the protein synthesizing machinery. So you don't have organelles that help reading the genetic code and then producing proteins that do all kinds of work for you, and enzymes that do all kinds of work for the cell. All they really contain is parts that are needed to actually get into the host, get into a host cell, and then to control the host. That's really what the bare minimum is what they have. And so virus particle is what really the structure is. Has a cover and a central core. So those are the two things. Really only has the core and the cover. And the core is a nucleic acid molecule, so it could be DNA or RNA. You've probably heard of DNA viruses and RNA viruses. That's a big distinction between different viruses, what nucleic acid molecule they're using. And then some matrix proteins, enzymes. They're not all found in all viruses, and they're not produced by the virus itself. So we will find out how they actually have, like how they get these different components. And then the cover is a capsid usually, and then an envelope. Again, that's not found in all viruses, and that helps distinguishing between different viruses. So the capsid, if you go back to the cover first, usually from the outside, the capsid is this structure that is really a protein and encloses the nucleic acid inside. So this blue structure is the capsid, and all viruses have that. Together, this whole thing together is called the nucleocapsid. Pretty easy to remember. There's a nucleic acid and a capsid, so it's a nucleocapsid. And as you've seen in the first, or in one of the first slides with all the different viruses, you have this capsid with, or this nucleocapsid, sometimes surrounded by an envelope or enveloped, and so they call these types envelope viruses, and these ones naked nucleocapsid viruses. Then the capsid itself is made out of protein subunits that are called capsomers. So that, and we'll look at the fine structure of that in the next picture. So there's the capsid, and it's made out of subunits of identical protein units that form these interesting shapes around the genetic material. So there's one, I don't know if anybody's heard of a icosahedron, but it's a certain structure that I guess has 20 sides and then 12 corners. and there's the helical type. And I think this one is not from your book. It's not really explained well because you kind of feel like, so you have these capsomers. These are the protein subunits that build this almost like this shape that if you connect a lot of them, you create these discs, and they connect to each other to produce this long cylindrical shape. And there's some viruses, and you'll see a picture of those where they really do look like that. So they basically just have this these capsomers attached to each other, and then the nucleic acid inside, and it forms this helix of capsomers, and you end up with a cylindrical nucleocapsid. And then the icosahedral shape is where you have 20 sides, and the capsomers look like this. So there's capsomers, and they produce shapes like this, and so that would be the face. And then you have these corners. So putting these two shapes together, you create this icosahedral shape, or icosahedron. And if you see, for example, in this case, that's a picture taken from one of those. It's a negative stain, so everything around again is stained, and the virus or the capsid is not stained, and you can see the shape of it and these fibers, or they call them spikes that are falling off, and we'll talk about what they do and why they're important in the next few slides too. So just remember, helical and icosahedral are the two main shapes of the capsid, and the capsid is really what encloses or what covers the nucleic acid, the genetic material. So those are essential parts of a virus. You will never, the DNA or RNA that the virus needs to take control over the host and make the host reproduce for it. So it's eventually a host, whatever cell it is, will take, or will be forced, if you want to call it that way, will be forced to use the viral nucleic acids to produce viral proteins. Then you always find these two structures. These two structures will always be part of a virus, so that it can reproduce in a host cell. And then in the cells that are not naked, where you have a little bit more complex, or a little bit more of a complex build, so you have the nucleocapsid, but you also have an envelope around it, and it has these spikes a lot of times. And you usually find those in animal viruses, and so in comparison, again, there's the naked one. So this would be the helix, and you can see this, for example, is a picture of one where you can see the shape of it is this long rod, and if you go into the fine structure of this one, you'd see the capsids and the way they're organized and arranged to build this structure, this virus. And usually the viral envelope is acquired when the virus leaves the host cell, so it takes part of the membrane of the host cell. So imagine you have an animal virus in a cell of a in some animal cell. When it actually leaves, when the viruses leave the cell and through different techniques, or there's different ways to actually exit the host cell, they take some of the membrane with them. and that actually is what produces this envelope. And then there are proteins, and those are the spikes. There are proteins that are included in this envelope, and they are then, since it's taken from the host, they're also important to reattach or to attach once that new virus leaves the host cell, to attach to another host cell. And some or all membrane proteins that you find in the host cell that was taken are actually replaced by viral proteins. So it's a pretty it sounds more complicated than it is, but it's a pretty smart way of viruses actually being able to get away without having to produce their own protein, which is so essential for any other cell. It's always so important to have specific proteins to facilitate a lot of the processes that are going on in the cell, to keep your homeostasis, to keep to get nutrients in, to break them down, to do all these different things. It's all to basically keep homeostasis and keep alive, and viruses are having found a way to let the host cell do that for them. This one is actually a figure or a picture of an influenza virus. So you have the nucleocapsid in there, and then you have this envelope with those spikes, which are mostly viral proteins. And just like with other microorganisms, the capsid and envelope are really there to protect the virus outside of the host cell. So for example, if you're in the host cell, if you're in the gastrointestinal tract, and the virus leaves the cell, there are enzymes in there, and so those enzymes, could be fatal for a virus if there wouldn't be any kind of protection through the capsid or the envelope. It also helps the virus bind to a new cell surface once it's reproduced and it finds a new, it's trying to find a new host. And then it helps penetration into a suitable host cell by attaching, but also in the process of getting to the nucleus. And here, for example, is herpes simplex virus, is a icosahedral virus. There are also, and I think the bacteriophages are important to remember, and they're easy to remember because they have this pretty interesting structure. The complex viruses, there's just some atypical viruses which are important in terms of diseases and infections that we care about, but they just have, in general, they don't belong to either one of those two groups, and they have they don't have the typical capsid, and they're covered by a dense layer of lipoproteins. But don't spend too much time. Just so you've heard it, and in case we cover one of those, they may not. I'll point out that they may not have the helical structure, or for example, like in this case, that would be a helical structure, and then again the icosahedral structure. So they might have a different structure. And then, as I said, the bacteriophages look more like this. They have a polyhedral nucleocapsid, and then they usually have these tail fibers and attachment fibers. So they look just a little bit more complicated than the viruses that are just composed out of a capsid head and the nucleic acid, or the ones that are enveloped. And there's so those obviously I pointed that out before, the bacteriophages parasitize bacteria. So a very small organism lives off of a very small organism, and we don't even see any of it. So now if you look at the differences, you have the complex viruses, which I said don't worry about them too much, but here you have a bacteriophage, and here you have that pox virus, the one, it's large, it's one of those atypical ones with the surface looking completely different than those. And then you have the envelope viruses. Again, there's the helical and the icosahedral over here. Mainly looking at the, and then you have here another couple. Which one is number 6? That's HIV. And then you have the non-enveloped, where you just have the capsid, and there's the helical and the icosahedral ones. And you can look, I mean, we can go through, we might, come across some of those during one of the lectures, where we look more into the differences and then what kind of disease they're associated with, or they're causing, and then we'll go into detail for those specific cases. So we've looked at this picture before. What would you say is the shape and envelope in this specific case? It's helical. Do you see any envelope on this one? So it's helical and naked. Very good. Couple more, 4 or 5 more minutes. So just to go from the capsid, which we just covered, so we looked at the capsid and the different ways that it's organized and arranged and the shape of it, to what actually is in it and what's protected, and that's the nucleic acid. And it's just like in every other living organism, it's the genome, it contains the characteristics, the genes to, or the hereditary material of that specific organism. And it's DNA or RNA, but never both. So you have DNA viruses, you have RNA viruses, but never RNA and DNA in the same one. So what those genes really do is they carry genes that are necessary to actually get into the host, and then to change the host's activity to make new viruses. So it's just to replicate that life form in a host. That's really what they're coding for. So the code in this DNA and RNA is mainly to get in and then to change the host to actually produce more of that and more of the capsid. So you have different numbers of genes, obviously, because they have depending on how complicated the capsids are, etc. You might need you need more information read by the host cell. So if you have a DNA virus, usually they are double-stranded, but they may be single-stranded, and the DNAs are either circular or linear. And then for RNA viruses, they're usually single-stranded, but there are also cases of double-stranded viruses. There are also cases where you actually have not a single strand, but segmented RNA RNA pieces in this capsid. And you might understand that better, maybe some of you already do, but the way the DNA is translated into RNA and then into protein, if you know that one, it makes more sense. All you have to know for right now is you can, if it's an RNA positive sense RNA, if RNA can be there can be differences. Some are immediately translated by the host cell, and some have to be converted into basically the opposite base sequence, and those are the negative sense RNA viruses. So this all you have to know right now is these ones, these negative sense RNA ones, there's an additional step before the genome can be translated by the host cell into producing the nucleic acids of the virus or the proteins for the virus. So really, that's the main message. At the minimum, viruses must carry genes for synthesizing the capsid and the genetic material. And then, so the virus gets into the host cell. Host needs, number one, they have to get in. Number two, they have to, and the host has to change their action to actually synthesize their genes, and then to package that material so the virus can get assembled and leave the host cell. So that's really what's contained in the nucleic acids of viruses. And with that, I'll let you go. And we'll do more viral work on Monday.